Okay. Good morning, everyone. My name is Lian Zhang from the Dermatology Hospital, South Medical University from China, Guangzhou. My topic is DNA methylation drives osteochondrogenic reprogramming in keloid pathogenesis and reveal therapeutic potential of decitabine. Keloid exhibits tumor-like characteristics and is also displayed in a hard texture with bone and cartilage-like changes, and also in get involved in the epigenetic modifications. This is published a lot of paper about this, and our teams published paper about the single cell sequencing as 2022 and reveal the trajectory of fibroblast differentiation into cartilage. When we found that the mesenchymal cells, ADM12 positive cells, and I found this group of cells exhibits a characteristic of this osteochondrogenic differentiation and characteristics. And we also did another research about the keloid, and we found that bone morphogenic protein BMP2 and BMP4 expression increased in keloid as well. And we found another osteochondrogenic gene, a related gene about the periostein is supposed to play a key role in collagen synthesis in keloid compared to the normal controllers. And this is this is paper published about the keloid exhibits bone cartilage characteristics. And this is published by other teams in the worldwide. It also exhibits characteristics of chondrogenic differentiation pathways. And our team also did a lot of research on the genetics, but we did very few positive results. Then return on the direction of the on the epigenetics. Epigenetics is the most biggest from their characteristic feature is the pharmacologically reversible. So, and we focused on the DNA methylation. And the importance of genetics in a lot of research did on the stem cells, the differentiation agent, cancer, and the keloid exhibits a tumor-like features, and that's why we think about that is that DNA methylation mediates osteochondrogenic differentiation keloid pathogenesis. Firstly, we do the several kind of sequencings about the transcriptome and the DNA methylation, DNA methylomes, and we found that we used the normal tissue and the keloid tissue and the keloid fibroblast and normal fibroblast. We found that compared to the normal tissue, the keloid tissue and the keloid fibroblast exhibits the DNA hypermethylation profiles. And the PCA data also show that this distinguish the phenotype between these three groups. And we also found that the keloid fibroblast showed similar results compared to the keloid tissue. And this showed that also higher the DNA hypermethylation profile. So we have a hypothesis is maybe the comparative analysis revealed the partially concordance DNA hypermethylation profile between keloid tissue and the fibroblast. And then we want to know we want to confirm that the keloid feature of osteochondrogenic differentiation. We used the keloid tissue and we do the RNA sequencing. We found that the volcano plot showed that the osteochondrogenic differentiation genes about a lot of like CDSK and the comp. These genes are the osteochondrogenic gene upregulators in the keloid. It also do the KEG gene pathway analysis showed that the osteochondrogenic gene, the pathway are enriched in the keloid tissue. And we also used the qPCR to verify this kind of results. We also similarly we also did the RNA sequencing in the keloid fibroblast. We also get the similar results, and we confirmed this kind of results used by the qPCR. Then we want to know whether the DNA methylation can to regulate the osteochondrogenic gene in keloid. Then we do the crosstalk between these two kind of the data sets. We used the DNA methylation profile, the methylome sequencing data sets, and the transcriptome data sets. We found that the group B. The group B is the DNA hyper DNA methylation and the upregulation of the genes. And this is happening in the gene body region, and the group D is happening in the promoter region with the feature of the DNA hypermethylation and the downregulation. And if the most of gene, the most osteochondrogenic differentiation gene located and happen in the group B. It means that this kind of genes have the like oncogenic features. And we do the crosstalk between these two data sets and we found that this DNA methylation hypermethylation controlled genes are the osteochondrogenic gene, and we also found that like BMP6 and this kind of genes upregulated, and we also did in the keloid fibroblast to get a similar result. Then we will this will make a small conclusion is keloid tissue and cultured keloid fibroblast exhibit a partially overlapping DNA hypermethylation profile. And then the osteochondrogenic differentiation pathway in keloid driven by the DNA methylation. Further, then we want to know another question. The core research question is, does DNA methyltransferase inhibitor then this targeting can show the demonstrate therapeutic effects for keloid by reversing osteochondrogenic signaling pathway? As we all know, especially oncologists know this kind of drugs. This is a very popular drugs for the blood disease. This is 2004 approved by the FDA for the MDS and other blood disease for about these two epigenetic drugs. These are this is the DNA methylation inhibitors for you. And we used this kind of inhibitors to find DNA methylation inhibitors to do the drug treatment because the keloid, the previous study we found that the keloid fibroblast can use this for the in vitro model for the keloid. And then we tested these drugs, and we found that even the low concentration drugs can show the promising effects in the cell proliferation and also in the cell migration. We used the DNA methylation inhibitor combined with another osteochondrogenic differentiation inhibitors, BMP inhibitors. We tested that it showed the synergistic effect, and also found that the Western blots show that this not only the DNA methylation inhibitor, but a combination treatment also showed promising effects in this osteochondrogenic protein inhibitions. Then we want to know the in vivo effects. We tested the drugs. We tested drugs in the PDX models. We embedded the keloid tissue in the mouse model, and then we generated the PDX model first. Then we tested these drugs. When we found that it showed a promising effect, the experimental group showed very small. The PDX models showed very small tissues and compared to the normal controls. Then we also tested the qPCR, tested this kind of osteochondrogenic gene also downregulated. Then we also do the muscle test about the collagen, and we used the serious staining also tested this kind of collagen in the postings. We found this kind of similar result that showed that these can inhibit the keloid collagen synthesis and the osteochondrogenic gene postings inhibition. Then we also made conclusion is DNA methyltransfer inhibitors suppress the cell proliferation by reversing the osteochondrogenic differentiation both in vitro and in vivo. Then this is the graphic abstract. We tested this kind of samples using the transcriptomes and methylomes, and when we used kind of these groups, and also used the outside the data sets published by other teams to verify this kind of results. And we found that the DNA methylation transcript, the methylation can control this osteochondrogenic features in keloid, and not only in fibroblast cells, but also in the keloid tissue as well. And also tested this kind of DNA methylation inhibitors. This is targeting to show that promising effects in vitro and in vivo. This is our team. We did a lot of work about the keloid, and our team currently have 22 members, and including a lot of the physicians, 19 physicians, or 6 of whom have associate professor higher titles, and integrating clinic medicines, engineering, and material science, and plastic surgeons, and the radiation, and translation studies. And this multidisciplinary research system can provide comprehensive technical support for keloid diagnosis and treatment and clinical research. We also published a lot of papers about the keloid research in during these 5 years, and also get a lot of fundings from the National Science of Natural from China and other governments organizations. This is also we also set a keloid biobank after 2023, and then from now and until now, the clinic cohorts is over 1,000 clinic patients, and over 500, 354 are the treatment naive patients. And this is biospecimens inventories as over 4,000 the FFEP tissues blockers, and over 4,000 RNA specimens, and over thousands the frozen tissue aliquots. And also did a lot of research, not just the basic science, but also the clinical translational medicines clinical trial. Okay, thank you for all. Hope we have the collaborative. Yes, thank you.
Yeah, so as Dr. Lian Zhang will go. Yeah. So since as we are preparing the next presentation, maybe we can ask Dr. Lian Zhang
some questions. Thank you for this nice presentation. Yes.
Yes. Okay, thank you for a question. This is a good question. Yes, we have photographed. We have a lot of photographs of every patients, and we have the like the clinic scale, like the VSS, VS, QID, or like that's kind of how to say in English? 评分 like the clinic score, and we said this kind of clinic cohorts. Yes, thank you. Another thing, so, and one of team members, she is doing the AI research. Because we have a lot of them, like over maybe 5,000 of the photographs, and we want to do the AI test to make sure how, for example, how many times of like injection, how many times of surgeries to can make the clinic outcome better, a clinic outcome. Yeah, we're doing this. Thank you.
So I'll ask you a question. You have figured out that there is DNA methylation problem. You have figured out that the cytidine, azacitidine possibly work. You have done it in cell models. You've done it in mouse models. What has stopped you from treating patients?
Okay, this is a good question. We actually wants to do this kind of investigator-initiated trial in our hospital, but this drug is a oncology related to the drugs, and because our hospital is a dermatology specific hospital, so we didn't have this drug in our hospital. I have to get this drug in our first hospital, then we can do this kind of, for example, after several lines of treatment failed, then we can use this kind of drugs for the patient first, because we consulted this kind of opinion to the other experts. They told us we have to do this for the patients. For this, the patients who have to recruit has to be used for several lines of treatments failed. Then we can try to treat these patients first. Then we can initiate this IIT clinic trial. Thank you.